Hello, everyone. My name is Maxine. I am an MD PhD student at the University of California, San Francisco. My pronouns are she, her. And as Sophie mentioned, I'm going to talk about how we would know what an astrocyte knows. And the first thing to talk about, I think, since it might not come up so much, is what an astrocyte is. So the brain, at least in humans, is composed about a third of neurons by cell. The other 2/3 are non-neuronal cells that we don't talk about a lot in neuroscience. The field is named after one particular cell type, after all. But it turns out that these cells have a really fascinating just physical architecture that suggests that they're incredibly intimately connected with neurons and their function. Astrocytes have a beautiful morphology, star cells, that was originally noticed on the basis of these individual projections that come out from the center of the cell, but they branch out from those main star-like projections into hundreds of thousands of individual processes that go out into what are called microdomains on the periphery. An astrocyte in cortex tile the entire cortex, so that each individual spatial segment, like a Voronoi tessellation, is covered by an individual astrocyte whose processes, microdomains, go out into the tissue. Now, astrocytes, as I said, constitute 1/3 of the brain. And some other things you might want to know about astrocytes are, without them, you die. So we can actually do this experiment. We can knock out with a machinery needed to produce astrocytes, and it is not compatible with life. And the other thing that we should probably know about them is that despite constituting a third of the brain, we essentially don't know what they do at all. In fact, I would go so far as to say, as similarly, that we might think of cortex as sort of being the control center for consciousness that has end effectors in the individual sort of like motor neurons that go out that sort of execute those actions, that perhaps we might even see astrocytes as the seat of consciousness operating on even longer time scales to do planning, and that changes the neuronal dynamics on a moment-to-moment basis. I don't mean this seriously, of course, but the fact that we don't know so much is sort of makes it suggestive and kind of interesting in that respect. So some things that we do know about astrocytes, though, are that specifically, if we think of individual neuronal synapses, pre and post, that in fact, the microdomains of the astrocytes up here in the periphery wrap around synapses, at a majority of them in cortex. And in these ensheathed synapses, these tripartite synapses, pre, post, and astrocyte, there are actually receptors that allow astrocytes to detect neurotransmitters and neuromodulators. And there is an ability for astrocytes, in addition, to release substances that change synaptic plasticity, including ATP and adenosine, and also to change neuronal excitability, including regulation of extracellular potassium and glutamate. So in fact, astrocytes sense and also affect neuronal dynamics in a very deep way. One thing that we do know about astrocytes is that, although we don't know what the action potential is, we are aware that if you look at them under a microscope under two-photon microscopy, over time with a calcium indicator, that there are these tiny excitations, these microdomain calcium events over time, that are heterogeneous in time and space and the quality of the individual excitation, and that have a very specific spatiotemporal correlation pattern, right? It's not just homogeneous white noise, but it actually has a very detailed structure that we're only just now beginning to understand the fine detail of. So in our experiments, what we did was we took a look at what happens when we do the system identification problem, right? So we took a slice of these astrocytes and we said, in one spot that's 10 microns across, we are going to, for 1 second, using two-photon photoencaging, give a pulse at either the neurotransmitter GABA or the neurotransmitter glutamate and see what happens. And it turns out that when we do this, that after a 1 second pulse of GABA, that there are events that are recruited in the astrocytes that flicker across the entire field for hundreds of microns away from the stimulation site. And this activation resonates for minutes afterwards. And in fact, if we instead do the perturbation with glutamate, we see a similar recruitment of astrocyte calcium microdomain events like this, but they are of a different character in terms of the actual spatial and temporal characteristics of the unfolding of the individual calcium excitations. In fact, they're so different that if we plug in the response of the astrocytes into a decoder and ask the question, was it GABA or was it glutamate? We can answer it. We can decode which neurotransmitter was uncaged in this simply by looking at the characteristics of the spatial and temporal extent of the calcium excitations that are recruited by the astrocytes after the perturbation. Now, it gets even crazier. This is results from our group that were recently published. You can look for Cahill et al. 2024. It was network level encoding of neurotransmitters in astrocytes. But other groups have looked at even some of the more canonical findings in neurons, right? So for example, if you have an arena and you have a mouse, as best as I can draw it, and the mouse navigates inside of this arena, right? And one of the core findings that we see in neurons in certain parts of the brain are that as a function across that arena, there are really distinct patterns of coding of neuronal firing. Some of them fire in a particular location in space, call those place cells, and some of them fire in a very distinctive grid-like pattern that tiles the arena in this way. We call them grid cells. And in fact, if you measure the calcium microdomain activation of astrocytes in a similar task, for astrocytes, there are in fact also place astrocytes. And similarly, although the signal to noise ratio is not quite so good as it is for neurons with respect to these astrocyte calcium fluctuations, because the imaging technology is not quite so good as we get with the voltage for neurons, we actually see that there are these timings of spatial activation patterns in astrocytes such that you can decode place information from the astrocytes of the mouse in the arena. In fact, you get orthogonal information coded in the astrocyte excitations that you get from the neuronal greater place field. So it's very interesting. It's very interesting to see these results of these types of information coding. But of course, this is ubiquitous in neuroscience, right? This type of Fourier-like decomposition of your stimulus is pervasive. We see it in place. We see it in vision, in Gabor filters that are present in visual cortex. We see it in auditory cortex with regards to frequency decomposition of sound. This type of representation of what's going on seems to be incredibly prevalent in multiple different domains. And so the question that arises for this, and also in artificial intelligence, right, where we see that there are in image classification tasks, Fourier-like representations that emerge, why is there such a universality of this type of representation across the tasks? So that set me on a rabbit hole, of course, trying to understand what might be underlying that. But I think, and though I won't say that this is necessarily a theory, or maybe even a framework, I'll say maybe I have a concept of a framework in the contemporary discourse, or a sketch perhaps. But it might be worthwhile to sort of think of it in the following way. So we know for a fact that mice or people, when they're present, or AI models, are able to learn some type of invariance with respect to the situation that they are in, right? So say I'm trying to learn something about recognizing a triangle. Well, there are certain things that you can do to a triangle, but the triangle still stays a triangle. You can reflect it across a particular axis, you can rotate it 120 degrees, and these things still keep the triangle as being the same triangle. So abstractly, there appears to be a structure, if we sort of just represent the objectiness of this triangle as this dot. There are ways that we can represent that there are transformations abstractly that we can do that have a particular structure on this. We can do nothing. Or we could reflect. Or we could rotate 120 degrees, say. And each one of these we can do to the object, stays the object. We can invert it, it stays the object. We can chain them together, it stays the object. And in fact, there's a wonderful field of mathematics that's aptly named representation theory, that actually describes that in any system that sort of is subject to these constraints of actually being invariant to these particular kinds of perturbations or modulations of the character, that, for example, if we look at the image in vector spaces, that systematically we can map the structure of that symmetry, of that invariance, onto corresponding endomorphisms, corresponding ways of being the same of the thing that is the representing object that we're looking at, in this case, a vector space representation. So for example, when we measure neuronal firing rates as a vector, the fact that the agent has actually learned to be invariant to this symmetry implies that there is a structural correspondence, just necessarily, no metaphysics, nothing else involved in this, just on the basis of the structural constraints of what it is to be invariant that's present in the representation. And in fact, we have even more than this. Because the representations are not just the representations, but the representations are in fact related to one another, right? So if I have, say, something that has symmetries alpha and beta, and I might describe one representation of this and another representation of this. In fact, I have a notion structurally of what it means for these two representations to be related. Right? If this is representation 1 and this is representation 2 that carries them through, then a transformation of those representations should be one that is consistent, such that if I move to one representation of this thing and then translate that picture over, it should be the same thing as carrying through by the original representation. And so we get an evident structure actually on our representations that there we can sort of just abstractly describe all of the structural relationships that are present in any such representation just on the basis of what is the structure of the underlying symmetry of the object. And we have even more than this, in fact. When our type of representation here, which we have as vector spaces, is particularly well behaved, which vector spaces, if you know, are familiar with linear algebra, actually tend to be fairly well. In fact, a lot of the times we can decompose a representation into individual constituent parts, right? We can say that this vector space representation of this that we have, I can actually really view it. One representation is kind of composed of two constituent parts that I can sort of glue together in a particular way, say for vector spaces as the direct sum. And what this corresponds to is the fact that in this sort of structure of the interrelationship of the different representational pictures, that I have a particular way in which I can always obtain this by sort of putting together these pieces. And similarly, I might be able to go down the rabbit hole of this one and say that there's, say this is 2A1 and this is R2A2 or something like that, right? But at a certain point, this ends and we get to sort of the prime factors of our representations that we have, right? And they're irreducible in some sense. Right? The individual constituent parts of this sort of can't be split any further and we get a unique decomposition of this, or at least a decomposition in some sense that we can't go down any further. Well, it turns out this is a really nice thing, the indecomposable representations of a particular symmetry algebra. In fact, if we say that that particular thing is, say, translations of three-dimensional space, it turns out that the indecomposable representations of that on, say, the complex numbers as our vector space for this, that is the Fourier transform, right? In fact, the unitary indecomposable representations on this, when if our notion of sort of sameness in a vector space is maintaining size, that those are in fact the indecomposable representations of the underlying symmetry being invariant to translation. I can recognize this image no matter how I move it. And in fact, even better, on the basis of those individual representations, and this is the beauty of it, you can actually reconstruct the content of the original symmetry that was present just by understanding, right, the individual Fourier components, by understanding what are the individual indecomposable representations. And I think that this is a really good view on sort of what it is fundamentally that we do in neuroscience, right? Like, there is some aspect of our system, of me, of the agent, that understands some concept of relationality, some structure that is present in the environment, right? I know what this chalk is no matter how I move it and rotate it, it is still chalk. And we see in the neuronal activation a representation of that that must necessarily be structured in a particular way. But it's just a projection onto something that we can measure. And the goal is to try and reconstruct, actually, the more abstract understanding, which is sort of substrate independent, which was the goal at the beginning, to try and understand, like, how would I go about taking these calcium excitations in an astrocyte and really seeing what they understand? So what's beautiful about this is that we can actually go even more abstract in sort of what is representational content, right, for a particular thing. So, for example, well, before we sort of thought about the case where there's sort of one thing that has sort of like some operations I can do to it that maintain its identity. Before we were sort of interested in the idea of the mouse that can move around in the arena. And the arena, okay, I can think of sort of all of the translations and rotations and things that I can do. It's still maintaining the arena. But there's a little bit more, right? Because there's sort of points that are present in this, right? We have a little bit of a finer relational structure that's present, right? Navigating the arena, I can go to a different spot in the arena, and it's still the arena, but it's kind of a different arena in this that's present, even though it's there, right? I can sort of imagine gluing individual ones of these sort of symmetry objects together into a larger picture. So this is what's called a group here with the one object with transformations for it mathematically. This is what's known as a groupoid mathematically in terms of multiple objects that are sort of linked together by sameness. And so the idea is that very similarly, we can generalize what the picture is of this abstract content to something that's a little bit richer, but still understand it in the same way by its representations in vector spaces, right? And so we see, in this case, we'll have multiple vector spaces that are all linked together by these transformations. And this gives us an evident structure of sort of understanding how we go about understanding a little bit of a richer relational picture. And similarly, I would argue that conceptual understanding in general can be seen on the basis of its relational character, right? So for example, I have in my mind, say, an idea of what chalk is, right? And it's in a relationship with the board, right? That's present in this way, right? It's in a particular relation. It's like writes on, say, my hand is also that. It's also here. It's something that I can represent and sort of, I know what are the transformations that I can do to hand that maintains its handiness. And one of those things is that it can come into relation with my concept of chalk, right? That's whole, say. And in fact, there's a sort of way in which my hand is therefore related to the board in this interesting and structured way vis-a-vis the chalk, right? I can sort of chain these two things together, like right, and then hold and then right. Sorry, we'll relate this compositions, then it's the order, right? So I can say like I write of the hold, and that's my relationship to the board, right? It's maintained through sort of the structural regularities of what chalkiness is. But very similarly, we can consider what it is to actually produce vector space representations of this type of object that maintain what is the structural pattern of sort of the abstract ideal of what chalk is, right? That's present in my own sort of mental picture of what's going on. I behave in a way that is concordant with my understanding in this relational picture. And so, you know, as an example of this is actually not a type of situation that's super unfamiliar, I think, in a lot of different fields. One I really particularly like is in the context of functorial quantum field theory. Right? This is actually sort of the object that is of study. Right? So we say, aha, well, I presuppose that there is some indexing object of information flow that I care about, right? In this case, it's the category of cobordisms, is what we say. Oh, I'm sorry, for vernacular situations, right? In this case, where I take my relational picture and I say, aha, they're not all exactly the same. Now the relationships are one directional. This structure is what's known as a category in mathematical sort of jargon. So the category I care about that indexes my information flow is given by sort of some topological spaces
And an arrow between them is given as a manifold that sort of connects the end pieces together, right? This is how I bookkeep like Feynman diagrams or something like that, right? Like two particles that are sort of interacting in this way. And they're linked in particular by coproducts, by gluing together in this particular way of adding disjoint unions. And what a functorial quantum field theory is actually exactly this picture. It is a vector space representation of it that preserves that product structure, right? And so what we get out of this is sort of a linear operator that represents this interaction, right? So this is a very familiar mathematical object in other disciplines. And in fact, as far as reconstruction is concerned, there's a wonderful result that's called Deline's theorem that actually shows that if you add a little bit of extra sauce into the upper picture of what you want for this, right? You say, okay, it's not just a topological space, but it's the space-like thing that you get from considering like algebras that you add a particular, like two integer symmetry on that maintains a braiding in a particular way. It's just an abstract nonsense way to describe it. But if I add some extra sauce into this thing that I say is my description of the information flow, then in fact, that corresponds to supersymmetry for the physicists, that in fact, this reconstruction problem is exact. I can exactly produce the character of the information flow precisely from these vector space representations. In general, that's not possible. There's a little bit of loss there, but in nice situations, it works out. So the last bit to say is that vector space representations are not the only representations we can do. In fact, we can say for this other one, well, I might care about some other type of data structure that I can measure, not just a vector of sort of neuronal activations. For example, I might care about representations in dynamical systems, right? So in this, we might say that our objects are sort of interfaces, right? I'll only draw this sort of picture for right here, one arrow, right? So for here, I might say that I have two objects and an arrow between them. That's a vector space or a representation of systems. Is a way of connecting in one direction, sort of the observations of the state, and in the other direction is responding to that state by providing an input in the opposite way. In sort of data science terms, we might call these a lens. So there's a way of characterizing this that that a few folks have done in polynomial functors. That's really sort of a nice way to get at that. So there's a vast generalization of these sorts of structures that's really nice. So to summarize and conclude, I think for all of this, I would say that what we really care about when we're studying these things is actually the overlying content that's present in the agent that's under study that we observe. Its invariance, its ability to solve the problem that has a particular structure. And we can actually characterize that very well mathematically by describing the relational structure in a language, the language of category theory, the language of these objects and their arrows. And so what we observe when we do scientific experiments, when we make observations, are in fact the representations of that overlying structure that preserve the structure, right? And in fact, there is a richness inside of what sort of representations that are structure preserving are allowed on the basis purely of the constraints that are provided by the structure. And in very nice cases, where we make it so that the information contents that we really care about, this sort of like ideal that's out there that we're trying to understand through its representations, is well constrained enough, for example, by our observations of agents that are able to solve particular problems that have symmetries, that in fact we can recover it on the basis of the representations that we observe. And with that, I will conclude. Thank you very much.